All right, so thank you everyone for the opportunity to give a small talk today. And I got really excited about the topic because it's something I feel very passionate about, and I'm glad to share passion with you today and some of the tools that we've been working in the Weiss lab and also I've been working myself. So I feel really excited about synthetic biology in general because I think it enables really precise control. Gene expression control, but it also enables spatial and temporal control of gene expression, and therefore you can control the developmental pathways that control development in general. I'm gonna show you what I mean in a second. In the Weiss lab, and I'm gonna overview a few of the tools that have been developed over the years in EBICS in the Weiss lab that are used to control and probe gene expression with the hope of precisely controlling emergence in organoid models. So these are some of the tools that have been developed in the Weiss lab over the years, and I'm going to highlight some of them. Before I get into that, I just wanted to share one of the reasons I got so excited about a synthetic biology approach to organoid. So if you look at around in nature, for example, in this case, we're looking at the way that a genetic oscillator, which is found in nature, helps to define the somites in mammalian development. It's a very, very, very beautiful system that is found in nature. And then in parallel, you see that in synthetic biology, people have also been able to engineer oscillators in living cells. You see things in development, like, for example, morphogens, so that's an important principle in developmental biology. And when you have gradients, then essentially you need to think the ability to sense concentrations, and also you need to be able to have the cells communicate with each other. So on the left, you have, for example, the morphogen gradient that helps define the neuronal tube during development. Then on the right, we have a set of tools that, for example, have been engineered and demonstrated in the Weiss lab to engineer cell-to-cell communication. And it is important in this case to have cell communication via along many ranges, so long range, medium range, short range. Besides just when you look at the field of synthetic biology, people have not only engineered cell-to-cell communication, but also, for example, the ability, and again, these abilities engineer them the ability to detect, for example, concentrations of specific molecules, which is some design principle that you also see in developmental biology in nature. And then obviously you see the other principle of developmental biology, the spatial patterning of gene expression. In this case, for example, we're looking at the spatial patterning of gene expression during development, which is very precise in a precise temporal and spatial fashion. And then on the right, we have an example of a circuit that was developed also in the Weiss lab, where you can engineer something called a cell state classifier, where essentially you can very precisely markers and the cell state in a population of cells and precisely activate expression only in one subset of the cells. So this allows you to potentially activate gene expression very precisely in a subpopulation, in a temporal and spatial fashion. Examples how we can apply this technology can be seen when we look at, for example, a platform that was developed in EBICS by the Weiss lab in collaboration with the Griffith lab. So again, many of you have seen this already. If you have not, very briefly, using lentiviral integration, they engineered a pulse of GATA6 expression that you can control using a small molecule. And you can engineer this in human-induced pluripotent stem cells. And then when you give a pulse of GATA6 expression to the cells, and then you just let the cells after that grow in basal, you end up with this brain-liver organoid, which has been highlighted many times in EBICS. Something important is that the organoid has also brain and liver tissue. So, for example, what would you do if you want to, for example, instead of having some of the germ layers developed into liver, what if you want them, for example, to turn into pancreas? Or what if you want the brain tissue to continue developing further? So how do you do potentially to reprogram this organoid? So central to this paradigm is the idea of sensing in the organoids. So it's the idea that you can sense a biomarker. So you can imagine that if, for example, you want develop the brain further, program the brain tissue to develop further, you want to develop a biological circuit that can be engineered in the stem cells, for example. And when particular cells turn into that brain tissue, you want to activate or repress gene expression there, but essentially you want the cells to be able to sense that state, have achieved that brain state. The same for any other developmental layer. So that is central to my work that I've been doing in Weiss Lab. And for example, this is a platform that we developed, and I have myself developed. The platform is meant to sense microRNAs. So microRNAs are widely found in development, differentially expressed during development. So you'll find different microRNAs expressed in brain tissue than the ones you find in stem cells. So you can use this differential expression to potentially have the cells sense when they have achieved the brain state. The way these microRNA sensors work, they have two transcriptional units. One of them, in this case, the MK1, which encodes for a red fluorescent protein, has target sites for a particular microRNA. You can change the microRNA based on what you want to sense. And then they also have a constitutive blue fluorescent marker. So essentially, in this case, if the microRNA is active or the cells are sensing the microRNA, then you expect to lose red fluorescence, but then they shouldn't change in blue fluorescence. So we've developed an RMC landing pad platform. RMC stands for recombinant mediated cassette exchange. We've integrated, we engineered this platform in the ABS1 locus in stem cells using CRISPR-Cas9. And when we integrated this microRNA sensor using this platform, we showed that the sensor is very stable over many generations of human-induced pluripotent stem cells. Not only that, but we could deliver, as you can see in these plots, on the very left plot, you have human-induced pluripotent stem cells that have been engineered with our microRNA sensor. Then at that point, we a microRNA that the cells can detect. So we're using a microRNA that is not expressed in human cells, so that we can deliver it and then characterize our system. And then in the middle plot, you see that after 8 days, the cells are losing red fluorescence, which indicates that this is human stem cells that have been engineered to sense this microRNA are correctly sensing the microRNA. And then by day 17, the cells have returned to their original state. So that shows that the microRNA sensor is stable. It was also able to sense the microRNA for which it was designed. Not only that, but we also differentiated the cells to human neurons using lentiviral integration of transcription factors, as you can see here. And we observed that the human neurons that we obtained from these engineered human-induced pluripotent stem cells fluoresced red and blue, which was indicative of the stability of the engineered microRNA sensor. We went also and used the GATA6 platform that I just mentioned to test also the stability of our sensor. So we infected the stem cells that had the microRNA sensor engineered in their genome with the GATA6 with two viruses, one of them which both of them which help us to engineer the pulse of GATA6 expression that I discussed at the beginning. We differentiated the cells in the same way, and we showed that after 2 weeks of differentiation and development, the cells still retain the microRNA sensor, which showed that the sensor was stable during differentiation, which is an important observation because in general, it's difficult to maintain long-term expression in stem cells, especially during long-term differentiation. In this area of sensing as well, the Weiss Lab has also recently shown that you can also have these embedded gene sensors. Essentially, there's a GATA RNA that can be expressed downstream of endogenous genes, and you can also sense them to, you can also use them to sense changes in gene. So similar to microRNAs, but you're instead of sensing microRNAs and looking for differential changes in microRNA expression, with this platform, you're able to sense changes in, you're able to sense differential gene expression, which is very powerful. Then so in the Weiss lab, the people had also engineered cell-to-cell communication, and we had discussed that there were also long-term, medium range, and platforms. Something also that was very relevant for development is the Optogenetic platform that was also in the Weiss lab and demonstrated in CHO cells. And this allows you, again, to have control, or to very precisely have spatial control over gene expression, which, as I mentioned at the beginning of the presentation, is a very important principle or rule that you see during development. And then another way in which actually also pattern, for example, organoids. So in the GATA6 platform, the patterning is an emergent property that happens naturally. But then in the Weiss lab, with work led by Jessie, she has shown that she can engineer differential cadherin expression in living cells, in human cells, and this leads to engineer patterning of the cells.
The videos work, or
no, they don't. Yeah, I tried. In terms of challenges with all of these circuits, and as we approach like the future of the field, we have shown, for example, with some of the efforts I've shown with the RMC platforms and CRISPR and so on, we have shown the ability to engineer single cells precisely. We can engineer sensors in stem cells, especially if the expression is constitutive. But then things are much more difficult for us to be able to engineer, for example, the instructions at the population-wide level. So we can engineer specific to have specific functions. That is much less clear how we can engineer population-level instructions, for example, for organoid development. Another challenge, obviously, is being able to engineer devices in human stem cells reliably that over long periods of time, especially during stem cell differentiation. And obviously, also one of the main challenges is that developmental biology is still a field that is not completely understood. So there are a lot of unknowns, and we don't know a lot of the pathways that we need to control in general and how development works in general. So with that, I covered my time, and I would like to thank everyone that I'm presentation, as well as our collaborators. And I would like to take some questions.
Thank you.
Sebastian, great presentation. Let me just kick it off with one question. You probably know that Ron and I have been working a lot on this P41 application, where the main contribution from Ron's lab is using optogenetics to pattern structures within organoids and cause, I guess, use that as a tool for producing new structures and within organoids, or induce different cell types within the organoid structures. How does that play into? How do you where do you see that going, and what potential do you see in that? In particular, I guess in connection with the first item I think that you had listed on your future directions.
Yeah, I think that area has a lot of potential. One of the ways I see that it has a lot of potential is because it merges computer-guided, essentially, control of gene expression with the biological tools that we're developing. So we're able to program, lasers and optics to control and pattern the organoids precisely. And I think that's a very powerful tool that is much more difficult sometimes to obtain just with the biological tools. Being able to, with a computer, very precisely pattern and control, especially gene expression. One of the main challenges might have to do with size. The light does a lot of penetration depth. So that might be a potential challenge as people try to engineer organoids. People are thinking of engineering them with low sizes, and obviously that will be a challenge if you try to use optic tool to penetrate the organoids in depth. There are some things that can be favorable, like for example, using far red lens that can penetrate a little bit more deeply. So I think yes, yeah, in general, a lot of potential because of this ability to control so precisely gene expression, not only temporally but also spatially. But then there are some challenges related to the optics and related to sterilization.
So Sebastian, I have a question related to this one. So when you talk about the spatiotemporal resolution with optics, currently is it at the single cell scale, or what's the scale are we talking about?
So you can potentially go all the way to a single scale, and I know that because people can do optogenetics at the single cell level. So I know the optic tools are there so that you can target a single cell. And also you can do them at the population level. A lot of the work that was done in the Weiss lab was at the population level. So if you go back to the slides to go into more depth, essentially this is a layer of engineered mammalian cells. And then the light is targeted using a confocal microscope to the region inside of this polygon. And essentially, only in that region, or mostly in that region, you see an increase in MK fluorescence, which is indicative of the ability of the light to activate gene expression precisely in that region of space.
I think you might need to consider the cell movement.
Yes.
So at the moment, this optical excitation still takes a long time. So in this case, it was 16 hours. And within the context of 16 hours, tissues can, there's some cell motility involved. And so if you wanted to just do it a single cell level, it is possible optically. The question is, how do you actually follow the individual cell to be able to make sure you're only exciting that cell as it moves around?
Unless there is some tracking algorithm in with it.
So if you do, you have to figure out how to track the cell, and then if that cell is not labeled ahead of time, then it might be difficult. One of the things that we try to do, for example, is nuclear localized fluorescence. And if you have nuclear localized fluorescence, then cell tracking is actually easier. So if you take an image every 10 minutes or so, then the nucleus hasn't moved that much, and you can actually correlate current nuclear location with this one, and that would allow you to do single cell tracking.
So what is light doing in this case? We have probably some light sensitive ion channels, right? That
no. So it's so the way the system works is that you're using two proteins, in this case CIB1 and CRY2HR. So those proteins dimerize when they're exposed to a specific wavelength of light.
Okay.
Yeah. So then what you do is that you can engineer, for example, you can fuse one of those domains to GAL4. In this case, it was fused to GAL4. GAL4 is a DNA binding domain. Then you fuse the other light dimerizable protein to a transcriptional activator. In this case, it was fused to BP64. So then when you have light, then the two light dimerizable domains dimerize, and then GAL4 can recruit VP64 to an engineer promoter, and then this engineer promoter is engineered using a minimal CMV promoter with upstream DNA binding sites for GAL4. For GAL4, in this case, 5 engineered binding sites, and then that gene is driving. That promoter is driving expression of MK8.
I see. So this protein would have to be within the nucleus, or it is in the outside the nucleus in the cytoplasm?
So
You shine the light.
yeah, so the protein can be in the cytoplasm. So for example, I believe the way this has been engineered, one of the 2 components has a nuclear localization signal. So I believe one of them is always in the cytoplasm, and
No, it has to relocate once it's bound. Once it dimerizes.
when it dimerizes, it localizes to nucleus.
Exactly.
Yeah.
Okay. Before dimerization, it's in the outside the nucleus in the cytoplasm.
Yeah. So
But again, this is a heterodimer. So there's actually 2 different elements,
yeah.
and then one of them is typically in the cytoplasm, and the other one shuttles back and forth.
Yeah.
And when they dimerize, then basically the mechanism, the one that shuttles back and forth, is able to carry the heterodimer into the nucleus.
Yeah.
Okay.
And you can also engineer the system so that both of them shuttle in and out. But generally, people that engineer the systems, they only shuttle one in and out, and the other one is in the cytoplasm, so that there's less leaky expression, because they find there's some off-target leaky expression when both of them are shuttling in and out of the nucleus.
Okay. Okay.
Can I ask one other question, I guess, before we move to the next ones? If he goes back a bit, with the GATA6 varying expression, I mean, you showed on the chart that high, medium, low are directed to different lineages. I was curious, and maybe now, maybe earlier it wasn't possible, but now, can you look to see how accurate that is? Meaning, how much of it is deterministic versus stochastic? So if you were to look at all the cells and what lineage they end up in, how many of them were either high, medium, or low expressors?
Yeah, so it turns out that it's not even like really high, medium, low. So we've been looking at this question a little more consistently and looking at GATA6 distribution levels. So what we hypothesize now, and again, we're in the midst of trying to understand that, is actually as cells go to the first iPSCs have low GATA6, then you have endoderm, which actually have a fairly wide diversity of GATA6 expression levels. The endoderm are then higher, and the mesoderm tend to be lower, but it's, there's overlap in the distributions. And then the hepatocyte, so the hepatic lineage that comes out of the definitive endoderm is actually lower, and the mesoderm, as they go to endothelial cells and they begin to vascularize, appear to be higher. So those trajectories overlap, and they also seem to cross each other. So there's a temporal evolution and amplitude evolution that are not monotonic.
But are you doing that with like one of the type of like CRISPR scratch pad systems or something, or just looking at those states? Are you doing it with like one of the CRISPR-based recording systems, or is that just looking at the relative levels of the different states of differentiation?
I mean, we're just looking at the GATA6 organoid and basically doing single-cell RNA across different days, and then correlating their overall lineage specification based on the overall profile of each individual cell.
Yeah, I don't know, I think it could be interesting because you could get at sort of like I said, the main question I was asking, deterministic versus stochastic, and how much of this in these systems, here you've got a tool for control of it, but do cells escape, and if so, at what frequency from sort of these overall rules?
Yeah, I mean, I think that's a very interesting question. We're actually interested in building these stochastic switches. So we actually have some ideas about how to build kind of lineage activated stochastic switches that may be able to push cells in different directions. But I agree. I mean, I think the stochastic aspect of it is super and not sufficiently well understood.
Well, thank you, Sebastian. With that, Ron, would you like to move on to the next talk from Avant?